Okay, so this week we're going to begin our science with an overview of the structure of DNA and RNA, and we're also going to talk about the central dogma of molecular biology. So we'll begin by talking about DNA. Remember that DNA is always going to be double-stranded. It looks like this sort of twisted ladder if you look here at the space-filling model, or you can see that here in the diagram. There are four main bases in DNA, and we represent those as single letters: G for guanine, C for cytosine, A for adenine, and T for thymine. G's and C's are always going to bind each other, and A's and T's are always going to bind each other. This complementary base pairing consists of hydrogen bonds. So again, between G's and C's there's going to be 3 hydrogen bonds. Between the A's and T's there's going to be 2 hydrogen bonds. This means that regions of DNA that are going to be very GC rich are going to be literally stuck together a little bit more tightly than areas that are very AT rich. And you can kind of see why. If they're stuck together 3 times, it's going to be a little bit harder to pull those 2 strands of DNA apart than if you only have these 2 bases, 2 hydrogen bonds holding them together. Now let's look a little bit more closely at the structure of these individual nucleotides here. They consist of a deoxyribose. This is a carbohydrate or a sugar backbone that is attached to a phosphate group. And you can see here in this backbone, the backbone consists of the sugar, phosphate, and so on, linked to one another. The bases, doesn't show the structure here, but the bases are a nitrogenous base. That means that they are going to have nitrogen in them. Nitrogen is going to be very important for life, and that is why. It's because it is located in the structure of the bases here. So moving on to RNA. RNA is primarily going to be single-stranded. Now we will talk about some examples where RNA is not single-stranded, is in fact double-stranded, but those are really the exceptions to the rule. RNA is really primarily going to be single-stranded. And of course that's in contrast to DNA, which is primarily going to be double-stranded. Another major difference between RNA and DNA is that it's going to contain uracil as one of the bases instead of thymine as its base. So instead of the G, T, A, C, in RNA it's going to be U for uracil instead. Another difference between them is this base is actually made out of ribose instead of deoxyribose. It's just a little bit different structure of the sugar, but you're still going to have this phosphate group that are going to be connecting them in this backbone. Also remember that there are 3 main types of RNA. mRNA, or messenger RNA, that is going to basically transcribe or be a copy of the DNA, travels from the nucleus out to the cytoplasm. tRNA is going to be responsible for transfer RNA is responsible for bringing amino acids to the ribosomes, and that's going to be really important for translation. The third kind of RNA is rRNA, and that is ribosomal RNA, and that is important for making up the structure of the ribosomes. So putting these 2 things together, we get the central dogma of molecular biology. That is, that DNA makes RNA, and the RNA is responsible for making protein. Said a little bit more distinctly, what happens is that genes are made up out of DNA. They're going to code for the sequence of nucleotides in the RNA molecules. They're responsible for directing the production of mRNA. The mRNA then leaves the nucleus, goes out to the cytoplasm, again assuming we're talking about a cell that has a nucleus, goes out to the cytoplasm. That RNA, that mRNA, is then read by the transcription machinery, or translation machinery, particularly the ribosomes. The tRNA is going to come in, it's going to bring the associated amino acids, and it is then going to bring about formation of a protein. And really that's the end goal of the DNA, of transcription, of translation. So another way to think about this is DNA is kind of like a cookbook that you have inside of your cells. In humans, we have 23 chapters, or 23 pairs of chromosomes. 23 types, 23 pairs, for a total of 46 chromosomes. Remember, you're going to get 1 set for mom, 1 set for dad. That's what makes your total of 46. Each individual gene on the DNA, on these chromosomes, could be considered a recipe. RNA could be considered the cook because the ribosome is going to be responsible for making the protein, and then the protein is really the end product that is going to be produced.